research programs. She has extensive experience in using quantitative cell biology to study the tumor microenvironment and cancer progression from a systems biology perspective. Her multidisciplinary research program focuses on providing new insight into the significance of the physical and cellular microenvironment of colorectal cancer progression through the development and utilization of physiologically relevant organ-dependent tumor models that allow for the characterization of cancer cell dynamics and serve as a platform for testing specific therapeutic modalities. The floor is yours, Professor Mumenthaler.
Thank you so much, Mumenth. And I greatly appreciate the invitation to speak to everyone today. Let me just pull up my slides. Hope my Zoom doesn't crash on me. Mumenth, can you see my slides in presentation mode? Okay. Perfect. Okay, thank you so much. And it's wonderful to be here today and give this webinar. Very excited to talk about some of our recent work that we've been doing, applying an organ-on-a-chip model to really study the progression of colorectal cancer. But before I jump into that, I did want to briefly touch on the Ellison Institute. So as Mehmet mentioned, I'm the Chief Translational Research Officer and faculty here at the Institute. And so we are actually neighbors to the Terasaki Institute. So we're located in West Los Angeles. But what's really unique about our institute is the fact that we are in I guess I wouldn't say it's brand new facility. We've now been here for a year and a half, but we have our clinic on the top floor of this institute. And so this is really where we see patients from wellness to cancer. And then we have our research facilities also within the same infrastructure. And our goal here is to really leverage technology to drive patient-centered research to enhance patient lives. And so we'd love to have people come and visit us. And so we're just really excited to be able to work with folks at Terasaki as well as others within the Los Angeles County and beyond. And so I do want to just kind of give a brief overview of what I'm hoping to capture today in this talk and really highlight how organs on chips or these microphysiological systems really can capture biological complexity. And so begin by some of the work I had. Actually, she was a grad student, a very talented grad student of mine who stayed on and is doing some work continuing this organ on a chip project. But she really spent a lot of time developing first this colorectal cancer on a chip model using cell lines. And then now we're really translating the model to mimic patient-to-patient heterogeneity using patient-derived organoids. And then I want to touch on how we've been integrating orthogonal assays to be able to measure cell behavior. So this really is integrating mass spectrometry-based metabolomics as well as on-chip live cell imaging to monitor the various different cell behaviors that occur within the tumor. And then ultimately, I think really to highlight how these organs on chip platforms are very tunable. And so we can study aspects of the tumor microenvironment and really perturb things such as the biomechanical as well as biochemical forces that occur within the tumor microenvironment and really show how this work has been able to lead us down a path of neurotransmitter signaling within the colon, driving invasive phenotype within the tumor cells. So that's really kind of the overview that hopefully we'll work on today and go over. But I wanted to actually really start by motivating the work. Why do we really need to have organs on chips to study aspects of the tumor microenvironment and cancer? And I think there was really a exciting paper that came out. Actually, it's now over 5 years ago in Science, where they took the eyelids of individuals and they sequenced the eyelids. And they actually found all of the driver mutations that you would find in cancer, yet these individuals did not have cancer in the eyelids. And so really, I think this just is a very simplistic study to highlight why you need this receptive environment actually for cancer to initiate and to progress. And so it really lends itself to understanding what is it about that environment that you may support these cells. And so also when we get into now thinking about the the transformed, as cancer is initiated and is progressing, we also look at the heterogeneity here. And really here, I think is also another simplistic image of showing two colorectal cancer patients. These are H&E images. These tumors actually have the same molecular alterations, yet they look vastly different, with the one on the right having a significant more amount of stroma. So this extracellular matrix and fibroblasts and other kind of desmoplastic type look to it, compared to the one on the left, which really is predominantly made up of epithelial cells. And so I think, both of these examples really lend themselves to saying that we need model systems within the laboratory that can help us recapitulate that microenvironmental heterogeneity, or that context of a patient's tumor. And if we can do it in a way that's tunable, then we can really see what the contributions of these microenvironmental factors might be on tumor cell behavior, both in the context of a primary tumor as well as tumor spread to the metastatic site. And so that leads me to kind of the work that we've been doing has primarily been in colorectal cancer. And I think, colorectal cancer is the second leading cause of cancer-related deaths in the United States. And while there's been a lot of work understanding the molecular alterations and kind of the stepwise progression in terms of transformation, there's still an unmet need in terms of when we think about the metastatic progression, and how the 5-year survival rate for these patients with metastatic colorectal cancer disease is less than 5%. And so we really need, better treatment modalities to address this critical issue in colorectal cancer. And there's also a lot of unanswered questions. We know that, colorectal cancer cells progress from the primary site, they intravasate into the vasculature, and then they're able to transit along under different forces, shear stress and others, and then extravasate into metastatic sites. And so there's a lot of kind of black boxes that we still don't know. And so how can we kind of develop a window into the missing pieces in the metastatic cascade? And so I think this is really where organs on chips have, I think, been very powerful. They're also, their utility is very broad and expanding. And so, the ability to really recreate these living organs with organ on a chip technology or these microfluidic based microphysiological systems are really designed to be able to model normal and diseased tissue level functions of human cells. And so we've seen actually a decade of innovation going from being able to model single organ chips and really being able to get the organ level physiology and functionality and toxicology assessment, all the way to now these interconnected organ chips to study PK/PD and drug screening and personalized medicine. And within that innovation, there's a lot of different factors that that the field has really been able to add to the complexity by engineering models that really can recapitulate tissue-tissue interface, or the biomechanical motions that are so important to a lot of these, to the organs and organ physiology, to really fluid exchange and the vascularization and innervation that is present within the body has really been exciting to see that. And now, how do we use these models to study different aspects of disease? And I think, I just really want to take a moment to highlight an exciting progress that was made towards the end of last year, where the FDA Modernization Act 2.0 was passed, where the FDA no longer requires animal testing before human drug trials. And I think this is a huge step forward in terms of animal welfare. And also, I think this ability to have microphysiological systems fill some of that gap in terms of being able to do drug testing, drug toxicity and safety analysis, as well as efficacy in model systems that are really human relevant and utilizing human cells. So excited to see really where the next 10 years of this field is going to go, but we've made significant progress, and now really turning our attention to in our group about how can we apply these systems to study human tumors, and to do this in a way to recapitulate that context. And as I mentioned, one area that we really want to apply them to is studying colorectal cancer progression. And so in particular, kind of that early metastatic spread. And so the ability for tumor cells to intravasate into the vasculature system. A lot of work in terms of other in vitro and in vivo model systems has been done before, and just laying out here kind of those different model systems, ranging in complexity from your transwell type system all the way up into your xenograft or animal model. And while all of these models are very important and have made progress in the field, I think there's definitely some advantages and also limitations to those more traditional models. And so here's kind of where we see organs on chips being able to resemble the in vivo tissue, to apply those mechanical forces, to have the ability to tune these various different microenvironmental factors. And then ultimately, I think the throughput is beginning to increase in these organs on chips. And so I think they're really going to be exciting to be able to apply to some of these questions that we have in cancer biology. And so to take a moment to kind of go through a bit more of the model system that we're using, and to orient you to the chip that we're working with. So we've been partnering with Emulate, which is a company that spun out of the Ellison Institute at Harvard. And so the Emulate organ-on-a-chip chip platform is a PDMS chip. And so here you can see that there's basically a cross-section of this chip. And so we have the top channel, which is where we're able to seed our epithelial cells after we've coated the membrane with an extracellular matrix. And then in the bottom channel is where we seed our endothelial cells to form this tube-like structure to mimic a blood vessel. And then you have fluid flow, and the fluid flow rate we can control in the top and the bottom channel. And then what's unique about this particular model system are these vacuum channels that are on the side of the chip, where we can induce cyclic strain to really mimic this peristalsis-like motion that occurs within the intestine. And so, really what we're trying to do is model some of the very unique and intricate aspects of the colon. And so we know that the gut is really a mechanically active organ, and that these mechanical signals, such as the shear stress that we find within the lumen, within the fluid flow from the lumen, as well as the cyclic strain from peristalsis, are really sensed by cells throughout the GI tract, and can actually coordinate GI motility. And so how this actually translates to the tumor is there's a lot of unanswered questions. And so having an organ-on-a-chip model that can recreate these mechanical forces and to actually model the progression of colon cancer and see how do these forces impact tumor cell behavior was really the question that we were trying to address. In addition, work that I'm not going to necessarily go into too much today is that we can add in layers of stromal complexity as well, stromal cell complexity. And so we have been introducing cancer-associated fibroblasts and other stromal cells into this model so that we can really see how the interaction of various different cell types happens over the period of time of these experiments, and really look at that coevolution of the tumor cells and the stromal cells. And so here's just to kind of give you an idea of how we set up these chips in terms of preparing and maintaining, and then the various different collection time points. One of the advantages, again, of organ-on-a-chips is the fact that they are the microfluidic capabilities is that we can collect the media, the affluent, and do various different measurements on the affluent, as well as measurements on the cells using imaging and other techniques. And so here's just to show an image of, again, using human intestinal microvascular endothelial cells, as well as we also work with human umbilical vein endothelial cells in the bottom channel to form that tube-like structure. And then in the top channel, some of our initial work, again, was seeding normal intestinal epithelial cells, and then seeding in colon cancer cell lines that are GFP labeled. And hopefully the video will play, which is this is going through the bottom channel. We have our endothelial cells through the porous membrane, now into the top channel where we have our intestinal epithelial cells and then our tumor cells there nestled in green. And so this is really to highlight that tissue-tissue interface. And so the ability to model the endothelial channel and the epithelial channel separated by this porous membrane, as well as in the epithelial channel, really showing the 3D nature. So we can form these crypt-like structures within the model to mimic the intestine. And so then what we can also do is not only do live cell imaging, and I'll show you some of that work that we've been doing to dynamically measure cell behavior, we can also take these chips and perform immunofluorescence and really look at specific cell type markers and do this more in a high throughput fashion using a chip imaging adapter and screening these chips on a high content imaging platform. And so really we can look at the length of the entire chip, the entire channel, both top and bottom, and look at particular markers of interest. And so here we're just showing initially, this is a lot of the validation work that we did very early on, which was, can we recreate the intestinal structure and function and really the different intestinal cell lineages within the chip. And so we can show here the different cell types, as well as also the barrier function. And so we were able to look at tight junctions using ZO1 expression and see that we could form these tight junctions within the chip, and also perform an assay where we basically add inulin FITZ into the top channel and then look to see how much of that is able permeabilize into the vasculature of the bottom channel. And really this helps us determine how intact our barrier is. And so we can show that both when we add our tumor cells or when we keep it just more as a normal intestinal epithelial chip, we don't see any effect on the barrier. We're able to maintain this intact barrier by day 4 of the experiment. And then one of the other things we wanted to do, as I mentioned, was to collect the affluent. And so we're collecting the media in both the inlet and outlet of these chips over time and looking at we're basically extracting the metabolites and really seeing how metabolites are changing over time, and then mapping those differential metabolites to the ingenuity pathway analysis. And so when we did this initially, we did this in both the epithelial affluent, which is what I'm showing you here, as well as in the endothelial affluent. But in the epithelial, when we compared our non-disease chip to 2 different colorectal cancer on a chip model, so these are, again, 2 different colorectal cancer cell lines, the HT29s and the HCT116s, the HCT116s being a more aggressive KRAS mutant colorectal cancer cell line versus the HT29s. And we see that in the non-disease chip over the course of 6 days, we don't really see a lot of change in the metabolites. However, when we add the tumor cells into the chip and then monitor that over 6 days, we see quite a number of metabolites changing. And when we map those differential metabolites to various different pathways, we actually see that colorectal cancer actually came up as the most significant disease state from these differential metabolites. And so this gave us some support and kind of validation that the model that we're generating within the Organoid chip platform is mimicking aspects of colorectal cancer biology. And so this was, again, a lot of the work that we did just to develop the model, characterize it, ensure that, we were able to see aspects of the biology that we were intending, and then to kind of adapt that model to really think about how can we translate this or move this model closer to patients and really to better understand the patient-to-patient heterogeneity and to be able to develop almost these patient-specific organ-on-a-chip models so that we can study different aspects of whether it's looking at efficacy of particular drugs or really understanding key aspects of the disease and the progression of the disease for individual patients. And so in order to do this, we really turn to our colorectal cancer biobank that we've been establishing over the past number of years. And here what we do is we're able to collect adjacent normal as well as colorectal cancer primary and metastatic tissues during surgical resection at the Norris Comprehensive Cancer Center. And so we're very grateful, again, for all of our clinical colleagues, as well as the patients that have generously donated their tissue for this work for research purposes. And so we actually, at our institute, we have a cell line team who really their charge is to ensure that we have high quality samples and models that we can develop from these tissues and ensure that it goes through the rigorous testing for quality assurances and really also collecting a lot of the metadata associated with these patient samples. And we work very closely with our clinical research team and clinical regulatory folks at the Institute. And so here again, when we get the patient tissue, we're able to generate organoids using the more traditional Hans Cleaver's method of generating the organoids, as well as isolating the cancer-associated fibroblasts from these same patients. So we really have this matched cell models available. We then expand and do a number of different testings to ensure the quality, and then do various characterizations in terms of RNA-seq analysis back to the comparing gene expression to the matched tissue, and then registering all of this into our LIMS system. And so taking this high quality organoid biobank, we initially started with 5 different patients, and we took these patients' organoids, and then we were able to digest them and seed them onto the chip, and then look at, under our 6-day experiments, as well as taking them out sometimes even longer than that. We really looked at how the cell is seeded, morphology differences, metabolomics changes, and then also gene expression changes. And so here's just to give you a snapshot in terms of some of the clinical data that we have from these patients. And as I mentioned, this is really kind of some of the earlier work. We're hoping to expand the number of patients that we can test, but you can see that we're able to really get a variety in terms of the sex, as well as ethnicity, and also the stage of the disease, and whether or not these are treatment naive, or also looking at the various different molecular alterations. And so this is really showing, taking those organoids and then fragmenting and digesting them into the chip and seeding those in the epithelial channel. And then we take our human intestinal microvascular endothelial cells, so the endothelial cells that are isolated from the intestine, and seeding those into the bottom channel. And this is really just showing on that initial day of seeding, what the chips look like. And then this is now imaging just looking at the top channel at 6 days across the different patients. And you can see kind of variations in terms of the morphology and the structures that are able to form, which actually this is the US patient, where it's actually a stage 2. So kind of earlier in the disease progression, you form actually more structures, more normal like intestinal structures compared to some of our stage 4 later stage diseases. Really, at this point, the disease has spread to other sites of the body. And you see here a much more disorganized morphology in some of these other patients. And then what we did, similar to the 2D cell line work, we also, again, wanted to make sure that within the organoids, the tumor organoids, we could generate an intact barrier. And so we performed the same inulin-FITC assay and really showed that by kind of day one of our experiment, we were able to form an intact barrier and maintain that across the rest of the experiment, as well as staining for our ZO1, our tight junctions, and then also in the top channel, and then V cadherin, looking at the endothelial cells in the bottom channel and showing the nice cell-cell interactions that are occurring. And so after kind of generating the model, we really wanted to study these physical forces within the intestine. And because, again, as I mentioned, this mechanical activity of the gut microenvironment is really important in the normal physiology of the intestine, but less is understood in the cancer space. And so really how do, these peristaltic-like motions, as well as shear stress from fluid flow within the lumen, influencing tumor cell behavior. And so here it's really a nice image of the intestine chip that Emulet has generated, and really showing the cyclic stretching like motion and also the normal intestinal architecture that can be formed within the chip. And so the first thing we looked at in our tumor organoid chip model is if we induce the stretching, the cyclic stretching like motion versus not, were there changes in gene expression? And so what we actually noticed initially when we did the PCA across stretched versus no stretched chips, we see that they cluster actually by the patient, which is not surprising given the level of heterogeneity from patient to patient tumor. But we did see changes in gene expression even within a patient between stretched and no stretch conditions. And when we map those to specific pathways, we actually, using gene set enrichment analysis, we actually see that the epithelial to mesenchymal transition was significantly increased in the stretched condition compared to the not stretched condition. And so, this started to lead us down this path of maybe these biomechanical forces that are present within the organ microenvironment change cell behavior and perhaps actually drive this more aggressive or invasive phenotype. And so that was kind of the beginning of some of the work where we then moved into investigating the actual cell behavior on chip. And so here we applied a live cell imaging workflow. And so what we're showing here is we imaged at 6 different regions along the length of the chip. And now actually what we're doing is the images I'm showing you here are really a cross section looking down the chip. And so you can see the red are the endothelial cells, and they're really forming this tube-like structure to mimic the blood vessel. The empty black space in the middle is actually where the fluid flow is occurring. The white dashed line is representing the porous membrane. And then above that white line in green are our tumor cells. And then the normal intestinal epithelial cells here are unlabeled, so you're not able to see them. And so here again, you can see nicely on day 0, the separation between the epithelial and endothelial channel. So you really get this nice tissue-tissue interface. However, over time, we start to see that tumor cells will invade through this porous membrane and then into the vascular channel. So by day 6, you still see like some of these individual cells invading through. And again, there's an extracellular membrane, a matrix, that's coating the membrane. And so these tumor cells are actually able to invade through that into the vasculature. And then some of them adhere to those endothelial cells. And then some of them end up into the affluent, which we'll show you in a few slides. So then what we can do is actually measure the number of tumor cells that are in the top channel, the epithelial channel, versus the vascular channel over time, and then actually plot really the invasion that's occurring, this invasion rate. So when we did this, looking at different chips, whether it was looking at just our more normal-like intestinal epithelial chip versus our tumor or colorectal cancer on a chip models with organoids or with tumor cell lines, what we find is that in the tumor on a chip models, we see more invasion happening when you have this cyclic stretching like motion or the peristalsis occurring versus when you really are in this more static environment, mechanical environment. And so, this goes and supports what we saw with gene expression changes, that you are starting to see the epithelial to mesenchymal transition occur. And then we're measuring an increase in the number of tumor cells that it can invade into the vascular channel under the cyclic stretching motion. So what we wanted to confirm, though, was, are we maintaining the barrier function in the stretch condition? And so, again, we did tight junction staining, like ZO1 expression, and we're able to maintain the formation in the presence of stretching, as well as doing the inulin-FITC barrier function assay and maintain that barrier whether you have stretching or no stretching occurring. What we wanted to also do was look at specifically the cells that had invaded into the bottom channel. And so we did some single cell analysis looking at cells in the top channel of stretched versus the bottom channel. And we actually see that they cluster pretty closely together, as would it be expected since they are the same cell type. But when we looked at gene expression changes between those and mapped those to different pathways, again, we started to see changes in EMT as well as metabolic differences between the cells that had invaded versus those that had remained in the epithelial or more primary tumor location. We also, as I mentioned, some of those tumor cells will invade and adhere to the bottom channel, and then others will actually flow into the outlet or into the reservoir where we can collect, and they're viable cells. So we can collect those, and actually they will expand up, and we can count the number of cells that end up in the reservoir, as well as measure various different changes that occur in those cells that are able to grow up. And so we term these circulating tumor-like cells. So we know that they are not CTCs, but really in this case, they are viable and they have invaded into that vascular channel. And so we really wanted to look at, are there changes in the cells that have been able to invade in a mechanically rich environment, so with this peristalsis-like motion present, versus when it's more of a static environment. And again, what we see here are a lot of changes in adhesion, as would be expected, and cell migration. But also one that we noted here was this GABAergic synaptic transmission. And, this got us starting to think about some of the neurotransmitters that we know are again, proven to play a significant role in the regulation of physiological responses within the gut, such as nutrient absorption and gut motility. But, really, how this might play a role in terms of cancer, and really thinking again about the gut-brain axis and that interaction, this started to lead down a new path that we wanted to explore. And so we went back to our metabolite work, where we're able to really measure these differential metabolites over time in the affluent. And we actually saw that those mapped the differential metabolites when you collected the media from a stretched chip versus not stretched chips, mapped actually to neurological diseases. And so again, we went a step further and said, let's look at it and really establish a neurotransmitter library within for the mass spec work that we were doing, and see, are there specific metabolite or neurotransmitters that are differentially measured in the stretched environment versus the not stretched. And again, one of our top hits was Gaba, and we see a quite high fold change of Gaba in the affluent in the stretched colorectal cancer chips compared to the not stretched, with a high significant p-value. So we went really to the literature to understand how much of this has been implicated in colorectal cancer, and there has been a couple of studies that have really occurred and shown that there's a relationship between GABA and colon cancer proliferation as well as invasion. But the in vitro evidence is a bit conflicting. And so, really what we were hoping to set out was, can the organ-on-a-chip model really help to elucidate the role of GABA? And especially because here we're seeing that this change seems to occur in the presence of these mechanical forces. And so in prior in vitro work, a lot of it is done in static conditions, and so we may not be seeing the same responses of the tumor cells. And so that is really kind of where we set off a lot of our investigations. And so one of the things that we wanted to look at, because again, we're seeing that there's a relationship between GABA secretion or GABA levels in the effluent in stretch condition. And so if we tuned this mechanical force, so if we were able to really do this intermittent type stretching, do we see changes both in the ability for tumor cells to invade, but also in the GABA levels? And so here what we're showing you is really the initial tumor cell invasion. So this is using our imaging assay where we're measuring the number of tumor cells that have invaded into the vasculature over time. And when we have complete stretching occurring the entire length of the experiment, you see, again, an increase in the number of tumor cells that invaded compared to no stretch. But then if you actually look at this intermittent, so where you have no stretching for the first 2 days and then you increase stretching, then all of a sudden you increase the number of tumor cells that invaded. And so we really see that these mechanical forces seem to modulate the tumor cell behavior. In addition, when we look at the GABA level changes, we also see that the GABA level tracks with that, where in our stretch condition for the entire length of the experiment, we see increasing GABA. But when we have no stretch, there's very little GABA. And then again, if we monitor like this green line where you have no stretch for the first 2 days, and then you add the cyclic stretching motion, you then see this increase in GABA levels. So it does seem that the GABA tracks with the mechanical forces that are present. But we still had a lot of questions about how much of these changes in GABA were coming from the tumor cells versus the normal intestinal epithelial cells versus the endothelial cells that might be present. And so we really needed to kind of tease that out. And so we looked at just, again, this normal intestinal epithelial and recognized that the KCO2 C2BBE1s are really not truly normal, but they do form more of a normal intestinal-like structure compared to a lot of the tumor cell lines. And again, here we see really no change in GABA levels when you compare stretching versus no stretching. But then as you look at our tumor or colorectal cancer on a chip models, you are able to now see that there's this differential level of GABA in the stretching. And then we also did a chip where we removed the endothelial cells. So we only had the epithelial cells present. And again, we see this differential change in GABA levels. And so this, all of this kind of led us to the fact that it's the tumor cells that really seem to be responding to the mechanical forces by leading to this increase in GABA in the effluent. We also did some QPCR arrays to really look at genes involved in GABA synthesis and GABA secretion. And we see again that these are elevated, not significantly, but elevated in the stretched compared to not stretched condition. And this was done through flow sorting of our tumor cells off the top channel of the organ-on-a-chip model. So then we also wanted to really look at the flux analysis. And so, really what is happening with GABA? And we think that there's this autocrine feedback that's occurring, but we then went and did some heavy labeled GABA as well as heavy labeled glutamine assays. And so here on the left, we took heavy labeled glutamine. We looked really at what happens then to, from glutamine and all the way to GABA, really the metabolic activity or metabolic flux. And we're able to show that in the not stretch condition, we see less heavy labeled GABA when you start with the label glutamine compared to the stretch condition. Apologies, these I realized are the same color, but so you basically see that there is this difference in the glutamine levels or the heavy label GABA levels in a stretched condition. And then what we also were able to show is that the intracellular GABA levels in the stretch condition were elevated. And so overall, this data is suggesting that the flux of GABA is increased by the stretched condition. And we're really working through additional analyses to understand exactly what is happening and how the cells are utilizing the heavy label glutamine in the stretched versus not stretched condition. But it did suggest that there may be this potential that cells are, tumor cells are actually metabolizing GABA and using that as an energy source for invasion. And so that led us to a couple experiments that we did on CHIP where, again, we took our no stretch condition where we get very little invasion happening. If we add just exogenous GABA, really this looking at the agonist effect, we see a significant increase in tumor cell invasion, even in the absence of stretching now, but because we've introduced GABA. But when we add mucimol, which is also a GABA agonist, but this GABA agonist doesn't actually get metabolized by the cells, what we see is that there is no increase in invasion. And so this kind of led us to the fact that maybe GABA is being metabolized by the tumor cells. And so we wanted to explore that further. In addition, we did look at the GABA receptor antagonists. And so in the stretch condition, again, we see just an increase in invasion of tumor cells when you have the cyclic stretching motion compared to no stretch. And then when you add the GABA receptor A antagonist, biculin, you see a reduction in invasion as well. So there's something occurring with the neurotransmitter signaling within the tumor cells. And so we also went back to our QPCR array and looked at Abat, which is, again, the enzyme that's able to metabolize GABA into succinate semialdehyde. And we see that is elevated slightly in the stretch condition. So then we went forward and really looked at Abat inhibition in our tumor cells. So we both knocked down Abat in the HCT116 tumor cells, as well as pharmacologically inhibited Abat. And when we look specifically at the knockdown expression, we're able to see that in the stretch condition, when you introduce HCT116 tumor cells that are knocked down for Abat, we significantly reduce the invasion compared to the stretch condition. And really, the Abat knockdown ends up mimicking more of the no stretched condition where we don't see much invasion. And then again, as I mentioned, we did the same doing pharmacological inhibition using vigabatrin. And when we compare from the stretch to and add vigabatrin, we're able to reduce the invasion. And it again mimics more of the no stretched condition where you see the minimal invasion. And so here's really where we're at the point where we think that is what is occurring is you have this peristalsis-like motion that is naturally occurring within the host milieu of the intestinal organ, and that these tumor cells seem to respond with increased secretion of GABA, and then an internalization or a metabolism of GABA by Abat, and that is promoting changes, these metabolic changes that are actually driving this invasion. And so there's still several pieces of this puzzle that we're working through, but this really highlights how developing this organ on a chip platform and being able to tune these mechanical forces when we wouldn't be able to do this in other types of model systems led us to kind of this new hypothesis in terms of neurotransmitter signaling and how this occurs really in normal physiology, but how tumor cells might be hijacking this signaling in order to further that invasive phenotype within the intestine. And so, this brings me back to kind of the initial part of the talk where I mentioned, there's several black boxes in terms of the metastatic cascade. And so a lot of the work that we focused on was really that first, early metastatic aspect where cells are intravasating into the vasculature. But ultimately, what leads to the mortality and morbidity of patients is really that metastatic spread to distant sites. And so that's where some of our focus is now is understanding, there's changes by these CTCs. We saw GABAergic changes in the tumor cells that were able to invade and ended up in the affluent, but are those cells able to extravasate into metastatic sites like the liver? And so very kind of early work that we're doing is using the liver chip model. And so this is really a quad culture model where we have hepatocytes. So it's the same base PDMS chip with the tissue-tissue interface separated by a porous membrane with now, the hepatocytes in the top channel, and then you have your liver sinusoidal endothelial cells, your Kupffer cells and your stellate cells in the bottom channel. And this liver chip can function like a liver. It secretes albumin. You get MRP2 expression. And we know that in colon cancer, predominantly the liver is the organ of metastatic spread. And so we are trying to understand those cells that are able to leave that primary tumor site. Are they able to extravasate and colonize the liver? And so here we can collect the tumor cells from the stretched versus the not stretched chips, and then take those tumor cells and flow them into the vascular channel of the liver, and determine whether or not they can extravasate and colonize into the hepatocyte layer. And so here we're able to just show, here's an image of the cells, the tumor cells that actually can extravasate and end up in the hepatocyte layer. And then when, number one, we wanted to look at whether or not these circulating tumor cell-like cells that we isolated could adhere to the endothelial cells and in the liver. And we see that the CTCs that were collected from stretch chips were able to adhere more to the endothelial cells than the not stretched CTCs, which also led then to more cells being able to extravasate and colonize the hepatocytes in the stretched condition versus the not stretched. And so really what we're trying to understand is this mechanical memory, or really this plasticity that occurs from the mechanical forces in the intestine, and how that might promote this more aggressive phenotype and ability to extravasate into the liver, and to note that the liver is a very GABAergic environment. And so really the link here between the neurotransmitter signaling of these tumor cells in the colon may play a role in survival in the liver. And so that is really kind of the direction that this work is taking. And so with that, I just wanted to really hope that through this work, we've been able to show how this tunable model system has been really informative in studying colon cancer progression and the tumor microenvironment. And I think we're really just scratching the surface in terms of the capabilities of these organs on chip platforms. And so I wanted to just also highlight one last thing. I mentioned it in the beginning, that when we are generating our patient-derived tumor organoid biobank, we're also isolating the stromal cells from the same patient, and in particular looking at cancer-associated fibroblasts, since we know that these are one of the predominant stromal cell types within the tumor microenvironment. And also, more recently, the literature really has been showcasing how heterogeneous the cancer-associated fibroblasts are. We used to think that mainly speaking, these CAFs were more of the bad actors, the ones that really contribute to disease progression and really this invasive and aggressive phenotype. But what we found is actually there seems to be different subtypes of cancer-associated fibroblasts, some that may restrain the tumor cell or tumor progression, and some that seem to promote. And there's also quite a bit of plasticity within these cancer-associated fibroblasts. And so we're really now exploring some of that on the Organ-on-a-Chip platform, and also understanding how it's not a one-way street in terms of cancer-associated fibroblasts influencing tumor cells, but really how do the other environmental factors within the tumor milieu influence the fibroblasts? So really understanding the mechanical forces present, and do those also contribute to activation or to some of this plasticity that we see within the fibroblasts. And so we've been able to collect fibroblasts across different primary colon as well as liver metastatic tumors, and do gene expression analysis, and then also look at the effects on CHIP, and both looking at taking conditioned media, so looking at the secreted factors and how that may promote tumor invasion, or physically seeding these cancer-associated fibroblasts on CHIP, and now doing it with our patient-matched organoids, and measuring changes in invasion. And so here's just some preliminary work showing quite a bit of heterogeneity in terms of the CAF response to tumor cell invasion. And this is, again, using that imaging assay that I walked through in the beginning, where we see that some of our cancer-associated fibroblasts seem to restrain tumor cell invasion compared to the tumor cells only. So compared to the control, some have no effect at all. And then some actually seem to significantly promote tumor cell invasion. And again, we see this both with the secreted factors from these CAFs, but also physically present on the CHIP. And so leaving you with that kind of on the horizon work, I really just want to take some time to acknowledge the team. It's been, very impressive hard work from a lot of students and scientists in my lab and across the Ellison Institute, as well as collaborators at USC and some of our partners. And also I really am very grateful for the funding by the National Cancer Institute, the Tissue Engineering Collaborative, as well as the Cancer Systems Biology Consortium grant. And at this point, I'm happy to take any questions.
Thank you for the fantastic talk, Professor Mumenthaler. Many questions came in. So one of them is, when you collect cancer cells from patients and develop organoids, is it difficult to grow them in vitro environment versus growing them within the human?
I mean, so we've spent some time optimizing the growth of the organoids, and a lot of this work has been published by other pioneers in the field in terms of organoid development. So we have especially with the colon cancer tissue, we have a pretty high success rate in terms of developing organoids. And so we're probably at about 90% right now that we can develop. Other organ types are more challenging. And again, there's a lot of optimization in terms of media components and other things. What I would really like to do, and this is kind of the next step, is to take some of the tissue that we collect and directly put those onto the chip. With now you have the fluid flow and mechanical forces from that initial state, which there you may be able to retain more of the heterogeneity of the tumor tissue, versus right now this step has been develop the organoid biobank, and then have enough material to then put those onto chips. So it's a bit of a stepwise progression, but the goal would be to go directly from tumor tissue to chip.
Thank you. On slide 20, the tumor cells are moving or dropping down to the bottom of the lumen instead of migrating to the side of the endothelial lumen top side.
I think referring to the cross section with the tumor cells that are invading through the porous membrane. Yeah, so they invade and actually crawl, some of them would crawl along the endothelial layer. And so that was just a single snapshot. Some actually stay at the top of the bottom channel. So they actually are on the, where the endothelial cells are at the very top of the channel. So we see kind of all different. I didn't show that. We've done some image analysis where we have a 3D reconstruction of exactly where the tumor cells are along the length of the channel. And so we get them both in the top of the bottom of the bottom, and they do crawl along the sides of the endothelial wall.
Thank you. Cancer affects vascularization. Are you planning on measuring barrier integrity of your model using tear sensors, in addition to permeability, tight junction proteins? How colorectal cancer can affect this barrier?
That's a great point. Yes, that is something that there's been some modeling that's been done on that, but it is challenging kind of in this particular chip setup. So agreed that is a limitation. We have not done tear analysis, only done the tight junction and inulin FITC assay.
Thank you. Have you looked at the expression of HLA-E in your model, as HLA-E expression and immunoinhibitory receptor is associated with colorectal cancer progression?
We have not. So that is, again, lots of things that are on our list to do, and especially the application in terms of immunotherapy and really combining, looking at whether we're isolating PBMCs from the patients too. That is some of the work that we're doing right now, is to introduce that into the chips, but we have not done that at this point.
Another question is, does it make sense to develop colorectal cancer on a chip that include gut microbiota, as it can have an impact immunomodulating the gut immunity as well as being connected with the gut-brain axis?
Absolutely. And I would love, yes, I definitely think it's something that comes up quite often. And I know my students are going, oh no, another area we need to add. But yes, that is exactly what we want to do. And it does make a lot of sense to include that.
Thank you. Thanks for the presentation. Have you checked the relationship between angiogenesis and Gaba?
Actually, yes. And there's some interesting clinical data that we've been exploring, looking at bevacizumab, so it's specifically looking at anti-angiogenic therapy and Gaba, and there is a significant relationship there. And so that is on our radar as well to better understand the vascularization and Gaba. There is quite a bit of a connection there. I don't have a lot of data that we've generated on it other than some of this clinical relationship that we've seen.
Another question came in. Does Gaba affect HUVEC membrane integrity?
Does Gaba affect what membrane integrity?
HUVEC membrane integrity.
HUVEC membrane integrity. I mean, so again, we're looking at when we do V cadherin staining, ZO1 staining in the bottom channel, so we look at tight junctions as well as the inulin FITC, we don't see an effect there. I guess maybe the question is asking more when we're looking at some of the antagonists too, or agonists. That's a good point. And I will say I don't have that level of data yet to comment if it's impacting the HUVEC. Or in this case, we're doing a lot more with the HIMEX, the intestinal microendothelial cells. And I don't think I have enough data to say one way or the other.
Thank you. What is the source of liver cells that was used in the chips? Are they available for others to use?
Yeah, so those actually come. So Emulet sells their liver chip as a bio kit, and so they source their hepatocytes. So these are primary hepatocytes that have been undergone a bunch of QC analyses, and so that is part of that chip. So we didn't source those separately. I'm not sure if they give the information specifically on sourcing yourself, but it is part of the liver chip model that they sell.
Thank you. Did you check the effect of glutaminase inhibitor regarding up and down expression of GABA?
We did not check specifically other types of inhibitors. We really initially were focused solely on kind of the GABA antagonists. Although again, we're thinking because it seems to be more of like the GABA transporters and then metabolism, there are some additional inhibitors that we need to look at. And I think that as we dig more into this GABA flux analysis, we'll be able to provide more insights there.
Thank you. Are there transcriptome differences between tumor cells in vivo and on a chip? Did you check it? Yes.
Stay tuned. No, that's actually data we have right now. So I didn't get a chance to really show it, but we have the tumor tissue, we have the organoids, and then we have the organoids on chip in stretched and no stretch condition. And so all of that analysis is happening right now to look at the relationship between the organoids that are on chip compared to organoid only, and then the relationship back to the tumor tissue as well. And so we do see differences there, but we need to do some more computational analysis to really be able to do that. But that is work that we're going to move forward with and try to make sure we publish that.
Thank you. Is it possible to make an immunomodulator organoid and test it in vivo?
And test it in vivo? That's interesting. Yeah, I mean, there are definitely ways to make the organoids and then to introduce those into mouse models. The question again becomes kind of the immune system and needing, if it's a human immune and then introducing that into a mouse. There has been some work done on that, but I wouldn't be able to comment too much on the success of that. But I do know that organoid to mouse, as well as humanized mouse models, have been explored definitely in the cancer space.
Thank you so much. That's all the questions I have. Thank you for taking the time for the talk and answering questions. Thank you so much.
No, thank you. Thank you so much.